Anatomy and Physiology, Chapter 12, the nervous system and the nervous tissue. So the main objectives of this chapter is to understand the anatomy and physiology of the nervous system, which is the master control system of our body. So we'll study the classification of the nervous system, the different divisions, the main functions of the nervous system, the different cells that make up the nervous system. They are called neurons and glial cells. We'll study them. We'll study the anatomy of the neuron. We'll study how neurons are classified and what type of neurons are found in different parts of the body. It's a cell that can conduct an electrical impulse. So we'll study, it's called action potential. So right now, a thought in your mind is nothing but changes in the ion permeability of the membrane. So it is called action potential. We'll study that. And we'll study how the action potential is generated and propagated down the axon. So the main function of this master system, it controls, it regulates and coordinates all the vital body functions. So it's the major communication system in your body. So it's vital for maintaining homeostasis. In other words, to stay alive, you need an optimally functioning nervous system. Its function is divided into three categories. One is sensory function. So the brain allows you to monitor your environment outside the body and inside the body. So the information that's entering the brain is called sensory input. It's usually through our senses, like your eyes, your ears, your nose, your gustatory, or your touch, through your skin, so your senses. So it's called sensory input. But your brain is also listening to your heart, your lungs, your urinary system, your reproductive system. So the message can either be outside the body or inside the body. So this is called sensory input or the sensory function. So your brain is constantly monitoring the outside and inside. So the information arrives at the brain, and here a very important function called integration happens. So this is where your brain processes that information and interrupts that information. Integration. It's trying to make sense of it. Then it will send out a command. For example, if you want to move your leg, your brain has to send a command for it to contract the muscles in your leg so that you can walk. So that is called motor output. So it's sending information either to muscles, sometimes it's also glands and organs. So they are called effectors. These are the effectors. It can ask a muscle to contract, it can ask a gland to either secrete something. So these are the three main functions of the nervous system: sensory, integration, and motor function. Coming to the classification of the nervous system. So if you take the nervous system, it is divided into the central nervous system or CNS and peripheral nervous system, PNS. The central nervous system, in this picture it's shown in green, includes organs such as the brain and the spinal cord. It's right in the center of your body, central nervous system. Peripheral nervous system are the nerves that radiate from the brain and the spinal cord. It can either be the cranial nerves or the spinal nerves. They will radiate out from the central nervous system and they will reach every tissue in your body. So under peripheral, excuse me, we have the somatic, which is the command that is going to your muscles, skeletal muscles. Soma means body. So skeletal system. Autonomic division of the peripheral nervous system will control your visceral organs and glands inside your heart, your liver, and so forth. It can either speed up the organ, then it is called sympathetic. If it has a calming action, it's called parasympathetic. So this branch of the peripheral nervous system, it will be covered in AP2. So sympathetic is your fight or flight response. When everything speeds up, your heart starts to race, you start to sweat. Whereas the parasympathetic is the rest and digest phase. So these are the main divisions of the central nervous system. So we'll look at it a little more closely. So if you take the central nervous system, which includes, the brain is made up of neural tissue, connective tissue, and a lot of blood vessels. It's a very needy organ. It needs a rich supply of ATP, glucose, for energy production. So as I told you, the main functions would be sensory, integration, and motor commands. Included in the integrative is also included higher functions of your brain, like your personality, your intelligence, your ability to learn, your ability to master your emotions, your memory. All of them are included in the higher order functions in the interneurons. Moving on to the peripheral nervous system, you can see it includes all the neural tissue that is found outside of the brain and the spinal cord. So these are the conduits. It's like wires that's going to take the information from the periphery of the body to your spinal cord and brain for processing. So it's going to deliver sensory information to the central nervous system, and then the brain will process it, and then it will take out motor commands. The incoming message arriving at the brain is brought in, it's called afferent division. So it's going to carry sensory information from the central nervous system receptors to the central nervous system. So it's going from PNS to CNS for processing. So that is called afferent. I would remember afferent as arriving at the brain, A. Whereas the opposite would be efferent, would be exiting the brain. So this is going to carry the motor commands from the central nervous system to the peripheral nervous system. So if you want your hand to pick up your pen, your brain has to send the command through the nerves that goes to your biceps to flex your elbow and do what you want it to do, and your wrist muscles. Afferent, arriving, efferent, exiting. So let's look at some of the main receptors. So like skin is studded with receptors. That's why you can, feel temperature changes. Or if somebody pinches, you would know it. If it's hot or cold, our skin is studded with receptors. These receptors can detect a change in the environment. And they will send the signal to if it's a reflex, it will send the signal to the spinal cord. It is processed, and then there's a motor command. So here the fingers have detected heat. So it's shooting up. There's no time to go to the brain. So the information is processed in the spinal cord itself. And the interneuron sends synapses. It's asking the motor neuron to contract your biceps so that you can pull your hand away and to safety. So these are called receptors. They are nerve endings that are found here. We have light receptors in your eyes, photoreceptors. So your ears have receptors to pick up sound waves. Effector is the response or the motor command that is coming, and here the muscle is contracting, so that would be the effector here. So it can be a tissue or cells. What are the two types of cells that you can find? So here's a micrograph of a neuron slide. So you can see a very large cell. It has all the different cell organelles, but it's highly modified because it's a very unique cell that is designed to transmit electric impulse called action potential. So the cells in the nervous system are called neurons. So this is a neuron. It has a lot of, it has a swollen part. It's called as the cell body. And then a long tail-like part called axon. But you can see surrounding it are helper cells. See the nuclei, they are called neuroglia or glial cells. So the two main cells that make up the nervous system is neurons that can send and receive signals, and they are interconnected. They are like information processors. And the glial cells, there's a lot more glial cells, like 10 to 50-fold more. And the glial cells are maintaining the environment for the neuron. They're making sure the neuron has the nutrients and the toxins are removed. So they will insulate the neuron, they will transport nutrients and remove the waste, and they will also mediate the immune response. It's like a nanny protecting the neuron. A group of cell bodies, so like, why, see, this is darkly stained, right? So a bunch of them together in the central nervous system is called as a nucleus. Whereas in the peripheral nervous system, it will be called as ganglion. Here it is, so that's why the gray matter, everywhere there's a bundle of cell bodies, it will appear gray in the microscope. So that's the reason for gray matter and white matter in the brain. Whereas the axons, they are called white matter. A bundle of axons in the brain or central nervous system is called as a tract. Whereas in the peripheral nervous system, it is called as a nerve. So this is a very important picture that you have to master. So a neuron, the anatomy of a neuron. So this is called as a multipolar neuron. It has all the components of a cell, but it is highly modified. So the structure is modified to meet its function, which is to conduct an electric current. So it has a swollen, enlarged part. It's called as the cell body. This is the cell body. It has a very large nucleus, and you can see the nucleolus inside it. So the nucleus will have the DNA, and the nucleolus will have the ribosomal RNA, and then it will have nucleoplasm and so forth. You can see that the cytoplasm is called as the perikaryon. Peri means like outside, karyon means outside the nucleus, so perikaryon. And it has mitochondria because the energy requirement is very high. And the cytoskeleton is called as neurofilament. You know, instead of calling it microfilament and microtubules, it will be called as neurofilament and neurotubules. They give structure to the neuron and the axon. So neurofibrils would be bundles of neurofilaments that is giving it support. These dendrites, these root-like fine cytoplasmic extensions are called dendritic branches. And this is the receiving part of the cell body. So it is going to receive the stimulus through the dendrites. So this is the receiving part, and the axon and the tails would be the output. So this is the input and that's the output. Endoplasmic reticulum has a lot of ribosomes. So it is very darkly stained. It is called Nissl bodies. Nissl bodies, that is rough ER, rough endoplasmic reticulum. It gives it a dark appearance to the cell body, Nissl bodies. There is one very long cytoplasmic extension. It is called axon. The cell membrane of the axon is called axolemma, and the cytoplasm is axoplasm. There can be branches in the axon. They're called collaterals. And then it ends in tiny branches called telodendria. And it's the telodendria will end in terminals called synaptic terminals. And this is where the synaptic cleft is. So here in this picture, the message shoots is this neuron is communicating with another neuron. However, they are not touching. So this is where is the synaptic cleft, and the terminal is called synaptic terminal. So this is the anatomy of a multipolar neuron. And anatomy is very important for us to understand in order to understand the physiology of the neuron. So as I told you, like here, one neuron is synapsing with another neuron. So the neuron that is before the synapse, or the neuron that is sending the message, is called as the presynaptic cell. And then there is the synaptic cleft. They are not touching each other. There is a gap here. And the cell that is receiving the message is called as the postsynaptic cell. So it is in the synaptic cleft where neurotransmission happens. So the presynaptic cell, when it is triggered, when the action potential is fired down the axon, it will release neurotransmitters that will find receptors in the postsynaptic cell, and it will transfer the action potential to the postsynaptic cell. Neurotransmitters are like chemical messengers that is released by the presynaptic cell. So the neurotransmitters are going to find, so here's the swollen end of a presynaptic cell, and then the action potential is coming down the axon. So when the action potential reaches the end, the synaptic terminal, the neurotransmitter is made in the endoplasmic reticulum. It will be packaged in Golgi apparatus. There will be an influx of calcium here, and the neurotransmitter, the vesicles will fuse with the cell membrane and spill into the synaptic cleft. When it spills in the synaptic cleft, it will find a receptor in the postsynaptic cell. So and then that will cause depolarization in the postsynaptic cell, and the message is transferred from the pre to the postsynaptic neuron. So the neuron can transfer the message to either to another neuron, or it can ask a muscle to contract, or it can ask a gland to secrete something. There are many neurotransmitters. Some main neurotransmitters are mentioned here, like acetylcholine. That's what we'll be studying in the next few slides. It is involved in muscle action and also in memory. Let's take another one. Dopamine. All these are neurotransmitters. Endorphins, dopamines, GABA, glutamate, norepinephrine, serotonin. This is the mood modulating neurotransmitter. And many drugs are designed to modulate, to either stimulate or depress this transmission according to the needs. For example, dopamine is for your mood, helps with sleep, helps with learning. So it gives you increased pleasure and suppresses appetite. I like that. Dopamine. What are the different types of neurons that can be found in our body? So depending on its structure, there are 4 main types of neurons. Anaxonic. So this neuron is very small, and then it has processes all around it that kind of look alike. It is found in your brain and in your special senses. Bipolar. So here, this neurons are also small, and then here's the cell body. On one side, it has the dendrite, and it has the axon on the other side. This can be found also in your sense organs, like your eyes, your nose, olfactory. Unipolar neurons have a very long axon. It looks like the cell body is pulled out, and it has like one long axon. A lot of fused dendrites on one end, and then the axon terminals on the other end. This is also found in sensory neurons of the peripheral nervous system. Multipolar neuron is the neuron, the anatomy that we just studied. It has a very long axon. It has a lot of dendritic branches on the cell body. And this is the most common, and this is found in your brain, in your central nervous system. So different parts of the body has these different types of neurons. So coming to the functional classification of neurons, I already told you sensory or afferent neurons. The neurons that, there's about approximately 10 million sensory neurons in our body. Whereas if you see motor neurons or afferent neurons, there's about 500,000. It is the interneurons or the association neurons. So here is in this picture here on the left bottom, you can see a sensory neuron bringing in the message. It is synapsing with the interneuron. So this is the processing neuron. And then the motor neuron or the afferent neuron will go to an effector in order to cause the action. So this is the functional classification of neurons. So these are structural, and now this is the functional. Oh, receptors. Receptors are also classified several ways, depending on the location. If the receptor is found inside the body, then it's called interoceptors, for monitoring your heart, your liver, and so forth. The receptors that monitor the external environment are called external receptors, like when you perceive temperature changes or touch. Proprioception would be the monitoring the position of skeletal muscles and joints, so it can monitor the position of your body in space. The receptor can either be a specialized structure and it is capsulated, or it can be a free nerve ending. They are also classified according to the stimuli received. If it is able to receive light, it's called as a photoreceptor. There's chemical chemoreceptors, temperature thermoreceptors, nociception receptors, and mechanoreceptors, pressure. So in the peripheral nervous system, we spoke about peripheral nervous system. In the motor neurons, it can either be somatic, that controls your muscles, or it is autonomic, that controls most your visceral organs. In this autonomic nervous system, we have the sympathetic, which is the fight or flight response, and parasympathetic, which is the rest or digest response. So this is the efferent division or the motor division. Interneurons are located in the brain, spinal cord, and in the autonomic ganglia between sensory and motor neurons. They are responsible for distribution of sensory information, for coordination of motor activity, and also your intelligence, your memory, your planning, your learning. All of them are residing in the functions of the interneuron. So we studied the neuron structure. We'll study how the helper cells or the neuroglia look like. So the neuroglia in the central nervous system, there's 4 main cells. And the neuroglia in the peripheral nervous system, there are 2 main cells. Ependymal cells will line the ventricles of the brain and the central canal and spinal cord, and they are the cells that is responsible for producing and circulating the cerebrospinal fluid. So this is the structure of an ependymal cell. Astrocytes, they are responsible for maintaining BBB, the blood-brain barrier. They provide structural support. They can regulate the ion and nutrient concentration that enters the neuron. They can recycle neurotransmitters. Oligodendrocytes are responsible for myelinating the central nervous system axons. So you can see how they can wrap themselves around the axon, thereby insulating it. This is important to speed up the action potential. Just like how electric wires, they have the insulation so that the electricity can stay in the wire. So that's called myelination. Microglia are the immune cells. They can remove the waste and pathogens by phagocytosis. So they will make sure the brain is clean and infection free. The cells that's found in the peripheral nervous system are satellite cells and Schwann cells. The satellite cells will surround the neuron cell bodies in the ganglia. They can regulate the oxygen, carbon dioxide, and nutrient content and the neurotransmitter levels around the ganglia. And Schwann cells, just like oligodendrocytes in the central nervous system, are responsible for this process called myelination. Myelination means insulation. It's responsible for insulating the axons, and also it can participate in repair. Here's more details, and there's another picture of each one of them. Ependymal cells are the cells. See, they have these ciliated cilia. Thereby, they can make the cerebrospinal fluid, and they can make sure it is moving, it is flowing inside all the chambers of your brain. It is made up of, it is shaped from squamous to columnar. It may be ciliated. They line the ventricles of your brain and the central cavity of your spinal cord. Astrocytes, remember, they are responsible for maintaining the blood-brain barrier. Very important. They will monitor the chemical environment of your brain. So only good things will be allowed to enter the neuron. It will stop any kind of toxins from entering the brain. Microglia is the immune cells that can eat up debris that's in your brain by phagocytosis. Oligodendrocytes is the cell for myelination. So myelination means wrapping around the axon, thereby insulating it. The two cells in the peripheral nervous system are satellite cells and the Schwann cells. So you can see here in the axon, the Schwann cell has wrapped itself round the axon, round and round and round, so that when the nerve impulse comes down the axon, it stays in the axon. So it's insulating the axon. So it's called myelination. Very important. So we have to next understand how the cell body is going to receive a stimuli and how it's going to fire the message down the axon that will result in the release of neurotransmitters. So it's all because of the transmembrane potential, the permeability of the neurolemma and the axolemma. So here, every cell is, when it's not stimulated, a neuron is not stimulated, is resting. So that is called resting potential. So the transmembrane potential is not triggered yet, so it's resting. But when it receives a stimuli through any of the dendritic branches, the resting potential is converted to a graded potential. So graded potential means a localized change in the action in the membrane potential. So the resting potential has received a stimulus, it's become a graded potential. If this is strong enough, then it will fire an action potential down the axon. It's called action potential. It is an electric impulse that is going to propagate down the axon in one direction, like that. And when it comes down the axon, when it comes to the synaptic cleft, then it releases the neurotransmitters here. It's called as the synaptic activity. So the neurotransmitters will find the receptor in the next cell or a gland or a muscle cell. And the transfer of information would be information processing in the postsynaptic cell. So these are the main steps in the creation of a transmembrane potential. This is a picture of a cell at rest. So this is what a normal cell looks like. So this is the phospholipid bilayer. And if you remember the fluid mosaic model of cell membranes, There's lots of protein channels. So when a cell, so this is the inside of the cell in orange, outside of the cell is shown in purple here. So there's these series of protein channels here. There's something called leak channel. And then you can see how the potash is like leaking. And then there's a sodium leak channel. So they are like leaking without, it's passive transport. Whereas the sodium potassium pump is a voltage gated active transport. So it will pump out 3 sodium ions and bring in 2 potassium ions. There's also some proteins that are in the cytoplasm that is negative. So overall, in a resting cell, the inside of the cell is negative compared to the outside of the cell that has more sodium and chloride here. So all these positive charges outside are because of sodium being pumped out. So positive on the outside and negative on the inside. So if you measure the voltage across the membrane, a resting membrane potential is negative 70 millivolts. So it's clocking in at negative 70 millivolts. So this is what we are seeing in a resting neuron. But when action potential, when it's stimulated, so what happens is the sodium will rush in. When sodium floods into the cell, it is going to become more and more positive. So here you can see negative 65. So this phenomena is called as depolarization when sodium is flooding into the cell. And then this stimulation will then lead to a graded potential. So as that depolarization happens, it picks up all along the axon. And the action potential is triggered down the axon. So the membrane potential is referring to the difference in the electric potential between the inside of the cell and the outside of the cell. The resting membrane potential is negative 70. When it receives a stimulus, the ion will flood in because the sodium channels are open. That phenomena is called depolarization. And when it becomes more positive, so negative 70, it's become negative 65. And then it will spread the sodium ions inside the plasma membrane that produces a local current that depolarizes the adjacent portions of the plasma membrane. So these are the different steps that are happening to the membrane potential of a neuron. Depolarization, so opening of the sodium channels. When sodium floods in, the cell becomes more positive, and then it will spread the action potential down the axon until there is a release of neurotransmitter. So the same thing in pictures here. So when an action potential is fired down the axon, the rough endoplasmic reticulum will make the neurotransmitter. In this case, it's a neurotransmitter called acetylcholine, ACH for short. It will package it in vesicles and it will fuse with the cell membrane and it will spill in the synaptic cleft. So you can see there's an influx of calcium when there's an efflux of acetylcholine. The acetylcholine, so calcium will enter the synaptic terminal, triggering exocytosis of acetylcholine. When acetylcholine finds the receptor, this is picture 3 here, the sodium channels will open. This phenomena is called depolarization. So when it is depolarized, the action potential has now jumped from the presynaptic to the postsynaptic cell. So after the neurotransmitter has done its job, acetylcholine is removed by an enzyme called acetylcholine esterase, so that it does not keep on stimulating the postsynaptic cell. So its job is done, so the ACH is removed from the synaptic cleft. So what are the different steps in creation of an action potential? So the first is the stimulus triggers the opening. So here's the resting voltage of a cell at rest, negative 70 millivolts. So when it receives a stimulus and sodium ions flood into the cell, so it's becoming negative 55, negative 45, negative 30, 0, and it can go up to like plus 30. So this is called depolarization. Sodium is flooding into the cell. All the sodium channels are open. Repolarization is from rapid inactivation of sodium but large efflux of potassium ions. So this is potassium ions are going out now. It's called repolarization. This will overshoot a little bit and then see it goes even beyond negative 70. It's called hyperpolarization. It's called refractory period. So you can't stimulate the neuron at this hyperpolarization. So this is by the efflux of potassium ions and closing of the potassium channels. And then it goes back to resting potential. So this is the action potential steps. Depolarization, sodium ions in. Repolarization, potassium ions out. Hyperpolarization is when it overshoots a little bit. And then it comes back to resting. So this picture is showing the changes in voltage or the electric impulse over the axolemma as the action potential moves down the axon. So this action potential moving down the axon can be either continuous or saltatory. So continuous can be seen in unmyelinated axons. Unmyelinated. So this is an unmyelinated axon. So there is a sequential opening of the voltage-gated sodium and potassium channels all along the entire length of the axon to propagate the impulse. So this is what like we saw right now. This is not as fast compared to the saltatory. So what happens in saltatory, we see this in myelinated axons. The action potential is propagated from jumping from node to node. So these nodes are called nodes of Ranvier because the sodium concentration gradient is moved on along the axoplasm inside. So it's very quick. So what have we learned during our time together? We studied that the nervous system is the master control system in the body. It's maintaining homeostasis in your body. It is made up of the divisions are the central nervous system and the peripheral nervous system. Central nervous system refers to your brain and your spinal cord. Peripheral nervous system are the nerves that come out of the CNS and go to the periphery of your body, to every tissue. It will innervate every tissue in your body. The main function of the nervous system is to the three main functions: sensory, integration, and motor function, all to maintain homeostasis or life in the body. We studied that the neurons can't function by themselves. The neurons are the cells that conduct the electric impulse or the action potential, whereas the neuroglia are the helper cells. Central nervous system has the ependymal cells, oligodendrocytes, microglia, astrocytes for the blood-brain barrier. So they're very important for the health of the nervous system or the neuron because they are maintaining the environment of the neuron. So they are the supporting cells of the nervous system. And we saw that the unique structure of the neuron, with a swollen cell body with dendrites, a very long axon. So the anatomy of the neuron reflects its very unique function to transmit a action potential. We studied the different types of neurons like unipolar, bipolar, and multipolar, and anaxonic, which are found in different parts of the body. Like we saw bipolar can be found in your sensory organs like eyes. We also saw how the resting potential creates a graded potential when it receives a stimulus. And then the graded potential will, after it reaches a threshold, will fire an action potential down the length of the axon. And that action potential will result in the release of neurotransmitters. And it is the neurotransmitter that will convey the message from the presynaptic to the postsynaptic cell. It can either be a muscle contraction, a gland that is going to secrete something, or another event. So those are that would be the effector activity. And I hope that was helpful.